I welcome you all to the second section of the first unit topic of physiology, human physiology. So the second section of the first unit titled as digestion and absorption. So here we are going to see about how our digestive system is usually digest the material what we ingest through the food, and how the materials which are present in the digested substances, digestive material, this gets absorbed. So we are going to see that in this particular presentation. And then first, if you see about the biomolecules, especially macrobiomolecules or important biomolecules, we usually say about three important components with respect to digestion and absorption. They are usually carbohydrates, proteins and lipids. And then one more biomolecule is that is nucleic acid, but their digestion is insignificant because the amount of nucleic acid is going to be very less. So we are not much thinking about the digestion of the nucleic acid, but we mainly concentrate on the digestion of carbohydrate, protein and lipids, and their absorption we are going to see as a major biomolecules. So in the slide you can see that how carbohydrates are mainly digested. So the digestion, the main process of digestion contains three important stages. That is like us degrading the macromolecules into smaller molecules, and then the smaller molecules are converted into biomolecules, simple biomolecules. Then the biomolecules are going to be absorbed. So the larger molecules, that is the macromolecule, or say the polymers. The polymers are first broken down mechanically in our mouth by means of mechanical grinding process. So after this grinding process, enzymes act on these materials and then they will divide that material into smaller molecules. So the major digestion of all the biomolecules mainly occurs by physical reactions that occurs in the mouth. Then the second phase of digestion occurs in the stomach, where the acid acts on the food material and it will also do some extent of digestion, and they are also activating the enzymes which are acting on this particular material, and they will also degrade most of the biomolecules, polymers, into smaller molecules. That occurs in stomach. Then later, after the mixing of pancreatic juice as well as the lipid digesting material which is going to come from liver through gallbladder, so they are going to also mix it with this material, that is partially digested materials, and then the remaining digestion is going to occur in the small intestine. So digestion is going to start from the mouth and it is going to end at the stage of, say, in an intestine. Okay, then similarly the absorption is going to start from the intestine and it also ends in intestine. But the intestine contains different parts like small intestine, large intestine, colon, that's all there. But the digestion is going to start from the mouth to the intestine, and absorption mainly occurs in intestine as well as it ends in the intestine, especially in the colon. So that is what we are going to see that. So this is the mechanically overall digestion process throughout our system we are talking about. Now within the slide we are going to see about how the carbohydrates are digested, absorbed, and lipids are digested, absorbed, and proteins digested, and how they are going to absorb. So the slide clearly indicates that you can see that amylase is the major enzyme which degrades the starch molecule, or say glycogen. If you are having an animal starch, that is called as glycogen. If the plant, it contains the starch. So both the molecules are partially degraded by the amylase which is present in the saliva. So the amylase degrades amylose and amylopectin part of the starch into maltose, maltotriose, and glucose oligomers and limit dextrin. So these are the four products can be obtained by the action of the amylase, which occurs in the mouth itself. And then when this particular maltotriose reaches the stomach as well as the intestine, the stomach and intestine we have certain enzymes like sucrases, maltases, isomaltases, and these enzymes are acting on these maltoses and sucroses and isomaltoses, and they will convert all these sugar molecules into a monosaccharides, that is the single sugar unit, that is the glucose form. So this is what the digestion part of carbohydrate. See that once the carbohydrate is digested, which occurs in the stomach as well as in the intestinal part, intestinal lumen, the molecule has to be absorbed across the intestine wall. So these things are carried out by, there are certain transporters which are present on the breast border membrane. Even the enzymes which are responsible for this digestion is also present on the surface of the membrane. You can see that isomaltase as well as sucrase are present in the breast border enzyme. Similarly, membrane. Similarly, the lactase is also present on the membranes. Similarly, there are transporters like glucose, sodium glucose transporters or glucose transporter 1, glucose transporter 5, and they are all present in the membranes, which are going to help for the transport of glucose after the absorption. So next slide, this slide clearly indicates that how this digested material, monosaccharides, are going to be absorbed through this intestinal epithelial cell. So that is what we are going to see from this particular slide. You can see that the digested fructose, glucose, galactose, say the fructose are absorbed through or transported through a glucose transporter, whereas glucose and galactose are transported through sodium glucose transporter, where the glucose molecules are transported inside the epithelial cell from the intestinal lumen along with the sodium ion, and from the intestinal epithelial cell the monosaccharides are transported to the interstitial fluid through the another transporter which is known as the glucose transporter. The sodium ion which comes to the intestinal epithelium is again transported by sodium potassium ATPase pump. In this reaction, sodium ions are moved to the intestinal fluid, interstitial fluid, and potassium is again transported back to the intestinal epithelial cell. The potassium which comes inside the intestinal cell is again transported back to the interstitial fluid for the next cycle of transport through a potassium channel. So this is how the digested monosaccharides, fructose, glucose, galactose, are going to be absorbed in our body. So this is what the digestion and absorption of carbohydrates. Next, the important molecule for the digestion and absorption is the proteins. So proteins are mainly digested by the acids as well as the enzymes, which are usually present in the stomach as well as the intestine. The stomach contains a protein like pepsin, whereas acid like HCl. The intestine is going to contain different enzymes which are transported from pancreatic juice to the intestine. So these enzymes are activated inside the intestinal lumen and they will act on the ingested material, that is the material what we have taken during the intake of food. For example, the enzymes which are going to act on the proteins are trypsinogen, chymotrypsinogen, proelastase, procarboxypeptidase A, procarboxypeptidase B. And these enzymes which are usually present in the zymogen form, that means they are actually present in the inactive form. Then they are converted into an active form by the action of certain enzymes or acids. For example, the enterokinase enzyme converts trypsinogen into trypsin. Then trypsin converts chymotrypsin to chymotrypsinogen to chymotrypsin, proelastase to elastase, procarboxypeptidase to carboxypeptidase A, procarboxypeptidase B to carboxypeptidase B. Then only these enzymes are going to act on proteins and they will cause the digestion of proteins. So why these enzymes are produced in a zymogen form? That is $100 question. Why they are produced in zymogen form? Because the body cells and tissues are all made up of proteins. So once the pancreatic juice, when it is released out of pancreas, the enzymes are in active state, then they will degrade the tissues as well as cells there itself. So in order to avoid the degradation of body tissues and cells, the enzymes are initially produced in the zymogen form. After the enzymes reach its target, that is what the intestinal lumen through this duodenal region, by the action of the enzymes, these zymogens are converted into an active enzymes and they act on the peptides and proteins for the digestion of proteins. So this is the formation of zymogen. The zymogen form actually prevents the digestion of a normal tissues. That is what you are seeing, the zymogen activation process. And the slide you can see that there are so many enzymes. Why we need the different enzymes? Because the different enzymes degrades the proteins in different manner. For example, if you see carboxylatase A, it removes the amino acids. For example, serine, which is present in the carboxylic end. Similarly, if you say carboxylatase B, it removes the amino acid arginine from the carboxylic end of a peptide. And if you see trypsin and chymotrypsin, they have an ability to break down the amino acids which are present in the amino terminal region is contributed by serine, that will be degraded by the chymotrypsin. Similarly, the arginine, that is degraded by the trypsin. So, depending upon the amino acids, different enzymes acts on the peptide. So, that is how all the proteins are degraded into a peptides and amino acids. So, here also you can see the proteins are considered to be as a biopolymer, and they are degraded into smaller peptides, and then they are degraded into a monomeric units, that is known as the amino acid. Then only the amino acids are absorbed, and to a lesser extent even small peptides can also be absorbed after the digestion process. So the slide clearly indicates that the small peptides can be easily absorbed by its respective peptide transporters, and amino acid is also transported with the help of the amino acid transporter, and the amino acids are transported along with sodium ions. So once the small peptides comes to the intestinal epithelial cell, inside the intestinal epithelial cell, they are broken down into amino acid by the action of the enzyme peptidases. After the conversion of the small peptides into amino acids and small peptides, that is the small peptides which are converted into amino acids, and already we are getting some amino acid from the digestion process. That amino acid is also transported to the interstitial fluid, and these interstitial fluids actually later form tubes like lacteals, and then it will transport the materials from the stomach, from the intestine to the liver. So that is where these amino acid reaches the liver through this lacteals and the arteries and veins. And then the sodium ion which comes to the intestinal cell is again transported to the interstitial fluid through sodium potassium ATPase transporter. Again, the potassium ion which comes to the intestinal epithelial cell again is transported back to the interstitial fluid through this potassium channel. So this is how the amino acids which are digested in the intestinal lumens gets absorbed. Similarly, next we are going to see about how the lipids are digested and how they are absorbed. So the lipids are usually degraded by the action of the enzyme known as lipases. So these lipases actually acts on triglycerides which contains glycerol as well as the fatty acid moiety, and then they will cut down this fatty acid and it removes the glycerols and fatty acids. Okay, then these fats can be converted into or emulsified into emulsified fat, and they are again acted upon by a pancreatic lipase enzyme to form a fatty acids and monoglycerides, and they are also get absorbed. See, the absorption, that is the digestion of carbohydrates as well as proteins are found to be very easy because they are lipophilic in nature. They are lipophobic, that is hydrophilic in nature. Once again I am repeating it. The digestion of carbohydrates and proteins are found to be very easy because our whole body is hydrophilic in nature. They are water loving components, carbohydrates and proteins. So they are dissolved in our fluid, that is in the digestive juice. They are gets dissolved, so they can be easily acted upon by the enzymes and they can be easily digested. But when you come to fat, the fat is hydrophobic in nature, they are lipophilic in nature, so they are never interact with hydrophilic, that is water molecules. So the digestion of fats has become very tough. So in this case we need a special molecules which can bring the digestion of fats very easy. So this function, this work is mainly done by bile salts which are going to be produced, which are going to be synthesized in liver and get stored in the gallbladder, and these bile acids in the form of bile salts are transported through the gallbladder to the intestine through the duodenum. So once this bile salts comes to this particular intestine, where the bile salts cause the process known as emulsification. You can see in the next diagram that the fat globules, that is the oils which are able to form a globe-like structure. If you place a few drops on water, you can see that the oils are form a round-like globes, globe-like structure. So this is known as the fat globules. Like that, when you took some oil materials in the food, It will also form like a globe-like structures in our stomach. So these type of globe-like structures which are formed by the oils are reacted with this bile salts. In the presence of phospholipids, they emulsify. Emulsification means they are broken down the big fat globules into smaller globes which are interacted by the bile salts as well as the phospholipids. So by attachment of this phospholipid as well as the bile salts, we are making the enzymes easily interact with the fat globules. So this is what the variations occurs due to this emulsification process. You can see that why we need bile salts as well as phospholipid, because the phospholipids and bile salts has both hydrophilic and hydrophobic part, because they are called as amphipathic in nature. So usually the molecules which contains hydrophobic means they are water hating molecules. The molecules which interact with water is known as the hydrophilic molecules. Vice versa, the molecules which will interact with the lipids are known as the lipophilic. The molecules which are not interacting with the fat is known as lipophobic. But some molecules will contain both lipophilic as well as hydrophilic nature. So such molecules are usually called as amphipathic molecule. So bile salts as well as phospholipids is one type, that type of molecule which contains both polar as well as non-polar region. So the polar region will interact with hydrophilic regions, whereas the non-polar region will interact with the fat. You can see that the formation of the fat droplets, which clearly indicates that the non-polar side of the bile salts will interact with the fat globules, as well as the polar part of the molecules will interact with the outer region. That is where the hydrophilic region is going to present. So this is how the big globules are converted into the smaller droplets. So after the formation of the droplets, after the attachment of the bile salts to the droplet, the enzymes which are going to act on this particular emulsion droplets will interact with this particular bile salt molecules. So this pancreatic lipases or other enzymes which will bind to this emulsion droplet by interacting with this particular polar region. So once the enzymes attach with this polar region, then they will start digesting the fat, and the fats are broken down into again micelles, and then the micelles are again degraded into glycerides as well as fatty acids, and once glycerides and fatty acids are formed, they are going to enter into the intestinal luminal cells. Okay, so once if it comes to the inside the intestinal cell, these molecules, that is what fatty acid as well as the glycerides, again combined with lipoproteins, and then they will form a molecule which is known as the chylomicrons, and these chylomicrons again enter into the interstitial fluid region, and then from the interstitial fluid region these chylomicrons goes. The lipid molecules are mainly digested by the action of phospholipids, bile salts, and enzymes like pancreatic lipases and other lipases. Okay, so they are acting on these lipid molecules, and they will digest them, the lipid molecules, into fatty acids and glycerides, and these glycerides and fatty acids, after its absorption through this brush border transporters inside the intestinal epithelial cell, they again come together to form a larger molecules, complex molecules, which forms a structure, specialized structure, which is known as the chylomicron. And this chylomicron is again transported to the liver through the lacteals. So now I will ask one question, that why the other molecules, if you see proteins, if you see a carbohydrate, they are converted from a larger molecular polymer into a monosaccharides or single units, and they will absorb and they will enter into the blood as such. Whereas the lipid molecules, after it gets converted from a larger molecule into smaller molecule, and then the smaller molecules are absorbed, and after absorption these smaller molecules are again converted into a larger molecules into a specialized structure like lipoprotein. Why you convert these lipids into a larger molecules in a specialized structure like lipoprotein? Why you convert these lipids into a larger molecule? This is because the whole body of our system is hydrophilic in nature, but these lipids are lipophilic in nature. So if you want to transport the lipids in a hydrophilic environment, you need a specialized structures like lipoprotein, which contains both the polar as well as non-polar part. The outer region usually containing the polar part, which interacts with this hydrophilic environment, whereas the interior part, which is hydrophobic in nature, lipophilic in nature, which carries the lipid into the liver through this lactease. So this is how the lipids are digested as well as absorbed. Next, we will see that other molecules, say apart from biomolecules like carbohydrate, lipids as well as proteins, the ions are gets absorbed through, there are certain specialized transporters which are present in the intestinal membrane. In the apical region of the intestinal membrane, we have several transporters which can transport the ions. Similarly, we have a special transporter which is known as the aquaporin. The aquaporin is used to transport water molecules. So if any molecules which is absorbed from this apical region and then the transported in the basement membrane, then the molecule can move across this intestinal cell. So this is known as the transition. The molecules are transported from lumen to intestinal cell and intestinal cell to interstitial fluid. So across the cells the molecules are transported. So this slide clearly indicates that the transport of ions like sodium, potassium, chlorine, bicarbonate ions, how they are all transported during this digestion absorption, and how the water molecules are also going to be absorbed. The most of the water molecules is usually absorbed in the large intestine, especially during the fecal formation. So that time only most of the water molecules are gets absorbed. So up to there we have the water absorption is found to be very less. So towards the colon region, we have more amount of water gets absorbed. And then the transporters which are going to transport the bicarbonate ion and chlorine, we have different specialized transporters are sodium potassium ATPase and chlorine transporters, and these transporters actually plays a vital role for the transport of molecules. So now you can see here in the tabular form of the enzymes as well as different parts of the digestive system where different enzymes and the molecules which are acting on the digestive process are listed. You can see that if you take a mouth, you can see that chewing is the important process, and there you have an enzymes like amylase and lysozyme, and they are involved in the carbohydrate digestion. And then usually in the mouth none of the compounds are gets absorbed actually. So this is how whatever we saw in the previous slides are summarized in the form of a table which can be easily understandable and remember. For remembering purpose, this might be the other way of representing the digestion and absorption at the organal level. Say what happens in the mouth and what happens in the stomach. You can see like that also the digestion and absorption. So we can see the digestion and absorption on the basis of the biomolecules, or you can study the digestion and absorption on the basis of the organs. So what happens? So that is also we can see. So that is what this table clearly indicates, organ-wise digestion and absorption you can see. Next you can see how the ions, especially the iron which is very essential for the formation of the hemoglobin, and it is also going to be a part of many important biomolecules. Say for example cytochromes. Okay, so they require iron for their function. So like that, several biomolecules requires iron for their function. So these ions are transported by different transporters are available. You can see that HCP is one of the transporter, DMT1 is another transporter, and these are going to transport the ions which are present in the intestinal lumen to the interstitial cell. So usually ions are transported in the form of heme or in the form of the ferrous form. So the ferrous ions are transported by DMT1, whereas the heme is transported by the HCP1. So once the ions are transported inside the intestinal cell, where these ions are converted into a ferric form and then stored in the form of ferritin, or they are again transported to the interstitial fluid by the FEN transporter. So this FEN transporter which transports ion to the intestinal lumen, where this Fe2 plus again converted into a Fe3 plus through transferritin. It is again transported through the blood to different parts of our body. So this is how the ions is also going to be absorbed from the digested material or the ingested material, and then it gets digested and gets absorbed. Usually only 10% of the ion we consume in the diet is actually reached our body. And this diagram, which clearly indicates that how much amount of solid material or the water which are taking in is going out of our body or it gets enter into our body. You can see that we are taking around 1200 ml of water per day and 500 to 800 gram of solids per day. But you can see the amount of substance which is released in the feces is only 100 ml of water and 50 gram of solids is excreted. So 10 times of the solids are usually absorbed. Only 10% of the material which we have consumed is released in the feces during the defecation process. Similarly, 1200 ml of water we are taking in average, only the 100 ml of water is released out of our body. Remaining amount of water, that is 1100 ml of water, is retained in our body. Apart from that, you can see that 1500 ml of salivary secretions are entered into our digestive tract, 2000 ml of gastric secretion come to the digestive tract, and 500 ml of bile, 1500 ml of pancreatic secretion comes to the digestive system, and 1500 ml of intestinal secretion comes to our digestive system. So if you calculate them, 1500, 2000, 3500, bile 4000, and you come to pancreas is a 5500, and intestine is a 600, 700 ml of molecules comes from the body to the digestive system. And then out of this 7000, they are all absorbed. For example, 6700 ml of material, which are ml of liquid material, which is absorbed in the small intestine itself. 1400 ml of material, that is the fluid part, is absorbed in the large intestine. The remaining 100 ml of water only, which is secreted in the feces. Similarly, apart from the digestive system, for example, mouth, esophagus, stomach, duodenum, small intestine, large intestine, rectum, anus, there are other organs like liver, pancreas, and gallbladder plays a vital role for the digestion. So if you remove the gallbladder or liver or pancreas from the digestive system, then the digestion won't occur. So this is how the digestion process as well as the absorption process occurs in our body. So whenever we took food, so we have to think about these processes which are occurring in our body. So if you have anybody have any questions, you can ask me or through mail. The mail ID will be sivabio@gmail.com, or if you want to contact through a phone, the phone number will be 9865866168. So through a phone, through WhatsApp, or through a mail, or through a Facebook, you can contact me to get clarified with your questions. Okay. The next, these are the book material which are utilized for the preparation of this chapter. So this concludes the second section of the first unit topic, that is digestion and absorption. Thank you.